There is a measurement most people have never heard of, and it takes exactly 60 seconds to run. In 1989, a research team at Yale University placed two strangers in a room and gave them one instruction: look into each other's eyes. Not at each other, into each other's eyes. 60 seconds of direct, sustained, mutual gaze. The kind that in ordinary life lasts no more than 3 seconds before someone looks away. What they found was not psychological. It was not a matter of feeling closer, of feeling seen, of feeling anything that could be easily named. What they found was chemical. Within 60 seconds of mutual gaze, the body had already begun a cascade of neurochemical events that, once started, could not simply be stopped by looking away. That cascade has a name, several names depending on which part of the mechanism you are examining. And the mechanism is why losing someone changes not just your world, but the chemistry of who you are inside it. The first name is oxytocin. Dr. Paul Zak at Claremont Graduate University in California has spent more than a decade studying what he calls the moral molecule, a name that undersells what oxytocin actually does. In experiments published in the journal PLOS1, Zak's team measured oxytocin levels before and after social bonding interactions and found that sustained mutual eye contact was one of the most reliable non-contact triggers of oxytocin release in the human body. Not touch, not speech, not proximity. The gaze itself to understand why you need an anatomy lesson. Most textbooks skip. Oxytocin is not produced in the bloodstream. It is manufactured in two small clusters of neurons in the hypothalamus, a structure about the size of an almond at the base of your brain, and travels from there into the posterior pituitary gland, where it waits for a signal to release. That signal can come from many sources: labour contractions, breastfeeding, physical touch. But it can also come from something far more subtle: the brain registering that another person is looking at you and that you are looking back. The eye contact does not produce oxytocin by itself. What it does is send a signal, a cascade of electrical and chemical events that tells the hypothalamus, this is a moment of social recognition. Someone sees you. And being seen for the human brain is one of the most ancient and fundamental safety signals in existence. The hypothalamus responds accordingly. But oxytocin is only the beginning of the chemistry. Beneath the oxytocin system, deeper in the architecture of what makes social contact feel like more than the exchange of information, there is a second system that most grief research has only recently begun to examine. It is called the mu-opioid system, the same system activated by morphine. The mu-opioid receptors, named for morphine because they bind to it, are distributed throughout the brain, but particularly dense in two regions relevant to eye contact and social bonding: the anterior cingulate cortex and the nucleus accumbens. These are the same regions that activate when you feel physical pleasure, when you hear music that moves you. Research by Dr. Tristan Inagaki at the University of Pittsburgh, published in 2015 in Social, Cognitive and Affective Neuroscience, demonstrated that social warmth, the feeling generated by sustained positive interaction with another person, activates these same mu-opioid receptors. Looking into the eyes of someone your brain has categorized as safe, known and significant does not merely make you feel connected in some abstract sense. It literally administers a neurochemical that shares a receptor system with opioids. This is not metaphor. The brain does not distinguish, at the receptor level, between the warmth of a known gaze and the warmth of a mild analgesic. Both bind to the same sites. Both reduce what researchers call social pain. Both, when withdrawn, produce something that functions at the neurological level, like withdrawal. Now consider what that means for the person who is gone. The research on this, on what the brain specifically does when that neurochemical source is permanently absent, is gathered in one of the most complete explanations of grief science currently available. It is called What Feynman Knew About Grief, and it is only available while this video is running. The link is in the pinned comment right now. If you have ever wondered why grief feels so physical, why it shows up in the body before it shows up in words, that is the resource that explains the mechanism. The link is pinned below. The third system involved in 60 seconds of eye contact, and the one that makes it different from 60 seconds of any other shared experience, involves a structure in the brain only conclusively identified in the early 1990s, and the discovery came from an entirely unexpected direction. In the laboratories of the University of Parma in Italy, a neuroscientist named Giacomo Rizzolatti was studying neurons in macaque monkeys that fired when the monkeys reached for food. A researcher reached for food while the monkey watched, and the neurons that would have fired if the monkey itself had reached for the food fired anyway. The monkey had not moved, but the part of the brain responsible for moving its hand had activated as if it had. These neurons were named mirror neurons, and the discovery that the human brain contains an analogous system, confirmed through fMRI studies at UCLA, Stanford, and the Max Planck Institute for Human Cognitive and Brain Sciences, fundamentally changed how neuroscience understands social connection. When you look into another person's eyes, the mirror neuron system in your premotor cortex and inferior parietal lobule does not simply register that a face is looking at you. It begins at the level of neural firing to simulate what it would be like to be that person looking at you. The mechanism is called neural resonance, a literal overlap of neural states. Studies at Princeton University by Dr. Uri Hassan, published in the Proceedings of the National Academy of Sciences in 2010, demonstrated through fMRI that during face-to-face social interaction, the brain activity of two people begins to converge, to produce similar patterns of activation in the same regions at nearly the same time. 60 seconds of mutual gaze is enough to begin this convergence, enough for your brain to start building a neural model of another person's internal state, their breathing rate, their emotional valence, their readiness. If you have found this useful, the explanation of what actually happens in the brain during connection and what the brain does when that connection is permanently severed. The link for what Feynman knew about grief is still pinned in the comments. It is only available while this video is running. Now, there is a layer beneath the mirror neuron system that most explanations of eye contact never reach. And it is the layer that makes what happens after loss so physiologically precise. It is the vagus nerve. The vagus nerve is the longest cranial nerve in the body. It runs from the brainstem down through the neck, the chest, the diaphragm, and into the abdominal cavity, touching the heart, the lungs, the gut. It is a bi-directional information highway, carrying signals from the brain to the organs and from the organs back again. And it is directly involved in what happens during eye contact. The neuroscientist Stephen Porges at Indiana University, in his polyvagal theory, demonstrated that the branch of the vagus nerve responsible for social engagement is anatomically linked to the muscles controlling facial expression, eye gaze, and vocalization. They are integrated. The same neural circuit that regulates your heart rate and your breathing is the same circuit that controls whether your eyes are open and receptive or narrowed and defensive. When you make sustained eye contact with someone safe, the myelinated branch of the vagus nerve, what Porges calls the social engagement system, activates. Your heart rate slows slightly. Your breathing becomes more regular. This is not a response you choose. It is a response the nervous system has been running since before language. The presence of a known safe face looking back at you is one of the oldest regulatory mechanisms the mammalian nervous system possesses. This is the mechanism behind what anyone who has lost someone will recognize but rarely describe in these terms. The specific quality of calm that came from being in that person's presence. Not from what they said, not from what they did, from the fact that they were there and looking. The vagus nerve registered it, the heart rate responded, the breathing shifted, and the brain filed it as a regulatory baseline. This is what regulation feels like. This is the reference point. And then the reference point is gone. The fourth system involved in 60 seconds of eye contact is the one that explains why its absence is experienced not as sadness, not primarily, not first, but as disorientation. Dr. Lisa Feldman Barrett at Northeastern University, whose research on the predictive brain has been published in Psychological Review and Current Biology, describes the brain not as a passive receiver of experience, but as a prediction machine. At every moment, your brain generates a model of what is about to happen, what you are about to see, hear, feel, and compares that prediction against incoming sensory data. When the prediction matches reality, the brain registers nothing. When the prediction fails, the brain signals an error. This error signal is what generates the felt experience of wrongness, of something being not as it should be. The link to what Feynman knew about grief is still pinned in the comments below, and it is only available while this video is running. Over thousands of interactions, your predictive brain has built a precise and deeply embedded model of that person. Not a general model of a human face, a model of their specific face. The way their eyes move when they are about to speak, the slight asymmetry in their expression when they are listening carefully, the quality of attention in their gaze when it is directed at you. The brain has practiced predicting that face 10,000 times, and 10,000 times the prediction has been confirmed. The model is not a belief, it is a neurological structure, embedded in the physical matter of the brain, the way a gravitational body occupies space in the fabric of space-time. The superior temporal sulcus, a region in the temporal lobe that specializes in processing the biological motion of faces and eyes, has devoted a measurable portion of its processing capacity to this specific face. Research from the Karolinska Institutet in Stockholm, published in Neuroimage, demonstrated that familiar faces activate the superior temporal sulcus with significantly greater efficiency than unfamiliar ones. The brain has literally become faster at processing this face than at processing any other. The neural pathways have been reinforced through repetition until they are, in the neurological sense, structural. When that face disappears, those pathways remain. The predictive model remains. The expectation remains. The brain, running its predictions as it always has, generates the signal. This should be here. The face does not appear. The error signal fires, and it fires again tomorrow morning. And again when you sit in a chair where they used to sit across from you. And again in a room where the light is the same quality as an afternoon you shared. Each time the face does not appear. Each time the brain signals. Error. Something is wrong. This is what is called yearning in the clinical literature on grief. Dr. Mary Frances O'Connor at the University of Arizona, whose research on complicated grief has been published in Neuroimage and Social, Cognitive and Affective Neuroscience, has described yearning as the central and most persistent symptom of grief. Not sadness, not depression, but the specific ache of a predictive system searching for something it was built to find and not finding it. The brain is not sad in the way language uses the word. The brain is running a prediction that keeps failing. And because the prediction system operates below the level of conscious thought, it runs even when you are not thinking about them. It runs when you wake up. It runs when you open the refrigerator. It runs when you walk into a kitchen at 7 in the morning and the light comes through the window the way it always did. The error signal fires. The face does not appear. There is a finding in the neuroscience of eye contact that most researchers move past quickly because its implications are difficult to hold. It concerns the difference between a live gaze and a recorded one. Your brain knows the difference, not in the way you consciously decide that a photograph is not a person, in the way that distinct neural circuits activate differently depending on whether the gaze is live or recorded. Research from the University of Trento in Italy demonstrated through magnetoencephalography that live eye contact produces a significantly different pattern of neural activation than looking at a photograph of someone looking at you. The superior temporal sulcus activates. The mu-opioid system engages. The mirror neuron network opens. But what makes live eye contact neurochemically distinct is not only the activation of these systems, it is the presence of what neuroscientists call temporal reciprocity. The fact that both brains are responding to each other in real time. The brain knows the gaze is contingent, that if you move, the other person's gaze adjusts, that if you speak, the eyes change, that there is another mind on the other side of the gaze processing you. No photograph carries this, no recording carries this. The brain detects its absence with the same precision it detects its presence. And so the photographs you return to, the ones you find yourself looking at, with a quality of attention that is different from looking at any other image, produce not the neurochemistry of connection, but the neurochemistry of something else. Something that initiates the systems and then leaves them without the signal they were building toward. The oxytocin cascade begins and cannot complete. The mu-opioid system opens and finds nothing to bind to. The mirror neuron network reaches for synchronization and encounters a surface it cannot resonate with. This is why photographs ache in a way that is distinct from other forms of absence. They are not simply reminders, they are the beginning of a neurochemical sequence. The brain was built to complete with a living person, and that now has nowhere to go. If everything in this video has made something click, about why grief lives in the body, about why the brain behaves as it does after loss. What Feynman knew about grief is still in the pinned comment below, and it is only available while this video is running. The link is there now. What remains when 60 seconds of eye contact is no longer possible is not simply an absence. It is the presence of a structure. The oxytocin system, calibrated over years to release in the presence of a specific face, continues to run its calibration. The mu-opioid receptors, trained to register a specific social warmth, continue to anticipate it. The vagus nerve's social engagement circuit, tuned to the particular frequency of one nervous system's co-regulation, continues to reach for the signal that was always there. The predictive brain, its model precise and permanent as a geological formation, continues to generate the face it was built to find. These systems do not dissolve when the person does. They are not capable of simply forgetting the face, the way you might forget a name. What was built over 60 seconds and confirmed over years is not an impression. It is architecture. It is structure embedded in the physical matter of the brain, in the synaptic weights, in the receptor densities, in the calibration of the autonomic nervous system. And structure does not simply cease, because the thing it was built to register is gone. The grief that shows up in the body, in the chest, in the difficulty breathing, in the strange exhaustion of an ordinary morning, is not separate from this neurochemistry. It is its expression. The vagus nerve is running a co-regulation protocol for a nervous system that is no longer present. The predictive system is generating expectations that cannot be confirmed. The mu-opioid system is waiting for a signal it received 10,000 times and will not receive again in exactly that form. There is something a physicist might notice here. Richard Feynman, after Arlene died, did not write about the chemistry of what he felt. He wrote about her to her in a letter he addressed and sealed and never sent. The letter was found in his personal archives 43 years after he wrote it, found only after his own death in 1988. It ends. Please excuse my not mailing this, but I don't know your new address. He knew where she had been. He did not know where she was. The letter was not confusion. It was precision. A physicist writing to coordinates he could no longer locate. Your brain, running its predictions, reaching for the gaze it learned to find, is doing something structurally similar. It knows what was built. It knows the face. It holds the chemistry intact, ready. What it does not have is the address. And that, not the sadness alone, not the absence as a fact, not the weight of the ordinary day. That is what 60 seconds of eye contact leaves behind. A brain that learned, precisely and permanently, to find someone. And that has not stopped looking.